small, quick reminder. If you have Slack up, I don't know whose Slack notifications those are, but just be careful to mute yourself or at least remove notifications from Slack.
We're live.
Should we get started then?
Yes, please.
Yes. Hi, everyone. Good afternoon, good morning, and good evening, wherever you are joining us worldwide. And welcome to the fourth day of the NeuroFemIndia 2021 conference organized by BiasWatch India. This conference is being run in collaboration with the ALBA network, which has generously agreed to sponsor the event, and our media partner in SciComm. I'm Bhavna Muralidharan, and I'm an assistant investigator and DBT Wellcome Trust India Alliance Intermediate Career Fellow at the Institute for Stem Cell Science and Regenerative Medicine, INSTEM, Bangalore. I'll be sharing today's MC duties with Dr. Sulagna Das, who is an assistant professor at the Albert Einstein College of Medicine, New York. And needless to say, I'm very happy to be doing this, and thank you, BiasWatch India. Both of us are members of the core organizing committee for this conference. And today's volunteers are Tasneem Mustafa, who's an alumna at IISr Pune, and Amartya Pradhan, who is a junior research scholar at TFR Mumbai, who will be helping us with the technical requirements of Zoom, the platform we are using today, and ensuring that we can have a productive interaction with our speakers and all of you, our audience. At this point, I would like to remind you that we are also live streaming this conference on YouTube. So for any members of the public and the Indian academia who've missed out on registering for the event, we'll be monitoring the YouTube chat stream in addition to the one on Zoom to select audience questions for our speakers. As you would have seen in our event posters for the day, we are doing public talks by 6 speakers, which is followed by breakout rooms from 4 to 4:30 p.m. India Standard Time. We have today decided to open the breakout rooms to any conference attendees who are interested in interacting more with our speakers. So anybody who's interested in networking, community building, and extended discussion, we'll be posting a Zoom link for the breakout rooms in the conference chat close to the end of our speaker talks today. You, regardless whether you have signed up previously or not, you are welcome to attend the breakout sessions today. And I highly encourage all of you to take advantage of this opportunity. After that, we'll be back here in Zoom webinar mode to host a very special session today, which is the Ungender workshop on gender awareness and bias sensitization. And as Abhilasha said a few days back, if you really want to be discomforted, please definitely join for the session today. So today is going to be a full and interesting day, and let's get right into it. I have 2 general notices for the audience. First is that the YouTube link will be provided in the chat. And then, as I said before, anybody who's not registered can use a link and access the conference. And we'll not be accepting any audience questions for the field overview talk, but for all other talks and sessions, we highly encourage the audience to ask their questions in the Q&A pop-up box. Please vote your questions, because if you want your questions to be asked and answered, please vote them, because we'll be taking up the questions with the most votes first. And please address your questions to the specific panelist by name. So please use the Q&A pop-up box. You can also put it in the chat box. Our volunteers will take them up. Right. So starting off today's series is Dr. Kavita Babu. Please join me in welcoming her. She's currently an associate professor and a 2020 DBT Wellcome Trust India Alliance Senior Career Fellow at the Center for Neuroscience, Indian Institute of Science, Bangalore. She previously worked at IISr Mohali, and she's been at the forefront of C. elegans neuroscience in India. She's also the recipient of the 2020 DBT Janaki Ammal National Women Scientist Award and the 2020 STARS grant from the Ministry of Human Resources and Development. Today, she represents the subfield of neuronal, cellular, and molecular biology, and will give a talk focused on the history and importance of this field. Kavita, thank you so much for accepting our invitation, and over to you.
Thank you very much, Bhavna. I'm just going to get my slides up. Are they visible?
Yes.
Yes, thank you. So first I want to thank the organizers for, sorry, it's not the first. The organizers for giving me this opportunity to talk about a little bit about the overview of the field and some of the scientists in India who are working on aspects of molecular and cellular neuroscience. And good day to all of you, wherever you're from. So I'm going to start off by giving you a brief overview of model systems and how model systems have been used for the past at least half a century to study aspects of molecular mechanisms that underlie neuronal functions. So some of the model systems that many of you have heard of would be worms or C. elegans or Cinorhabditis elegans, flies, Drosophila melanogaster, and fish, Daniel Rerio, and rodents and mouse musculus. I'm going to talk a little bit about principal investigators who set up their labs in the past, who've had their labs for more than a decade in India, who work on the top 3 model systems. And I'm going to start off with the model system that I work with, which is C. elegans. And just to give you a brief overview of C. elegans, Sidney Brenner was one of the first people to start using model system, or one of the pioneers of C. elegans biology. And he started using worms as a model system in the mid-1960s. So with that, you can just understand that the field is fairly new. It's only been for the past 50 plus years that you've had researchers working on C. elegans. C. elegans has 302 neurons, and everybody knows where each neuron is and who each neuron is making connections to, making it a really interesting system to start studying the molecules and signaling processes that occur in this system. Apart from that, humans have approximately 25,000 genes. C. elegans has 20,000 genes. Many of the genes are conserved, genes required for postsynaptic receptors, making neurotransmitters. All of those are largely conserved in C. elegans, making it a really good model organism. C. elegans has been used to study the molecular mechanisms underlying cell death. And this was work by Horowitz and his co-workers. And a lot of beautiful neurobiology in C. elegans has been done by multiple labs, including that of Corey Borikman, who's looked at molecules and circuits in C. elegans. India has not had too many C. elegans labs till recently. The first lab was set up in India, working on C. elegans was in the 1970s. And then there was a huge lull for a very long period of time before the next couple of labs started in the mid-2000s. I'm going to talk about one of those labs. The second model system, which is what I worked on during my PhD and which I'm still extremely fond of, Drosophila. This is just a nice quote by Will Blake. And Drosophila as a model system has lots of advantages in that it has a brain, a lot of neurobiology can be done, a lot of behaviors can be studied. A lot of Drosophila neurobiology in India was set up by Obaid Siddiqui at TIFR. And there have been a slew of labs, and there are still a lot of young people who are setting up their labs with using Drosophila in India. And I'm going to talk about a large number of Drosophila labs in the coming slides. Drosophila as a model system was set up by Thomas Morgan and his colleagues, and there have been lots of interesting discoveries using Drosophila, including Hox genes, which are required for patterning in all organisms. The last model organism that I'll be talking about is zebrafish. Zebrafish was started as a model organism by George Streisinger, and in the 1970s. Again, just giving you a brief idea that all of this is happening fairly recently. And it was a lot of work by Jan Jonas and Walhart that allowed one to start looking at patterning in zebrafish. And multiple labs use zebrafish to study aspects of neurological diseases as well as neuronal development. So with this, I'm just going to talk about some of the PIs in India, or all of them are women. And Shubha has already spoken about Veronica, but I felt this would be incomplete if I did not speak about a little bit about Veronica as well. Veronica has been my mentor. I've done a summer project in her lab many, many years ago. And she has been an outstanding mentor. So Veronica has, as all of you would have heard yesterday, worked on Drosophila. And she's done some beautiful studies which have looked at circuit development in Drosophila. I'm just going to take you through one small study where she looked at a proneural gene. So this was work done by Tanisha Zaveri. They've looked at a proneural gene called atonal, and they found that this is required for the formation of glomeruli in the Drosophila olfactory lobe. So you can see that in wild-type conditions, the axon of a neuron that expresses atonal forms these nice connections with the olfactory lobe. In the absence of atonal, these connections are lost. This is just a very small snippet of the real large body of really beautiful work that has come out from Veronica's lab. And apart from just doing really beautiful biology, she's also been an outstanding mentor. She was the head of the Department of Biological Sciences at TI for multiple years, and I found that she used to take interest in even the summer students project. So she used to talk to us about our work almost on a weekly basis. So she was like a mentor par excellence. She also set up multiple schools that allowed students from Cuba, Iran, South Africa to get access to modern neurobiology and meet scientists. So I wanted to start off with her because she's been a huge role model in my life. The second person I'm going to talk about very briefly about is Geeti Hassan at NCBS Bangalore. Again, a fly biologist. And Geeti did her PhD from Cambridge and a postdoc with Mike Ross-Pash's lab, where she looked at aspects of circadian biology. In her own lab, she looks extensively at calcium signaling and the role of intracellular calcium stores in fly neurons. She's mentored multiple students, and many young researchers who have gone through her lab now have their labs at different parts of the world. She's also currently the vice president of an Indian National Academy, INSA. And I've had multiple interactions with her, and I remember she visited IISc Mohali once, and all of my students at that time met her with presentations, and she very patiently and enthusiastically sat through each one of them and gave them really, really good suggestions. Some of her recent work, which is shown here, essentially looks at how the state and properties of dopaminergic neurons are regulated by IP3 calcium signaling, and how this in turn allows for changes in the flight behavior of flies. She's also recently started working with rodent models and has recently published on Purkinje neurons in rodents. The third person I want to talk about is Sandhya Kaushika from TIFR Bombay. Sandhya did her PhD from Brandeis, and she worked with Drosophila, and then moved to Washington University St. Louis to work on C. elegans neurobiology. So she made the nice switch from flies to worms. And her lab is largely interested in studying trafficking of cargo vesicles and axons. And they use multiple techniques that allow them to study how cargoes move. Just as an example, recent work by Parul in her lab has shown that stationary cargo and actin can come together to prevent normal movement, of a smooth movement of cargo that's mobile cargo. So her work essentially deals with is very heavy on cell biology, where she looks at multiple proteins and gets mechanistic and insight into how they function in axons. Apart from this, Sandhya has also been a mentor to all of us who have been C. elegans labs in India. So Sandhya set up a lab in the mid-2000s, and she really has truly been somebody who's helped all the younger labs that came on later. So she was the organizer of the first two young YIMs, Young Investigator Meetings, which many of you would have gone through. Apart from that, she also started the first Indian worm meeting and the Indian worm PI meeting, both of which have been invaluable for me, and I'm sure they've been invaluable for everybody else in the C. elegans field. So she's somebody who's brought a lot of us together in the C. elegans field, and because of her, we have a sense of community. And I think this would not have been possible without the amount of work that she's done. She also has, more recently, she's been involved with science communication with respect to COVID, and she's involved in consortia that bring details of what COVID does to the public. So she's also collaborated with many of us younger PIs, and I'm so grateful to have had her as a mentor when I first joined, and now as well. So essentially, C. elegans in India, she's really been a huge mentor and has really been a cheerleader for all the labs that have joined in the past few years. And then now there are a bunch of us, and we really know each other and meet each other virtually in life, in real life as well, thanks to the effort that she's put. Another fly lab in Banaras Hindu University, Varanasi, is Mausimi Matsudi, and she works on signaling on the signaling molecules involved in development of neuroblasts. Her lab also works on rodents, and they're also interested in looking at genome sequences of Indian populations. But largely, they started off as a fly lab. Beena Pillai is the first zebrafish lab that I'm going to talk about. Beena did her PhD in IISc, and right after her PhD, she moved to IGIB, where she's had her lab for the past decade and a bit. Her lab is interested in studying in looking at genes that are expressed in zebrafish neurons. So they look at long non-coding RNAs that are expressed in neurons and how they function. They also do a lot of genomics experiments and use zebrafish as a tool there as well. Shiba Vasu has had her lab at JNCSR, again works on Drosophila. And her lab is interested in looking at circadian rhythms in Drosophila, and they look at how different aspects of different conditions like changes in temperature could affect circadian rhythms. Anuradha Ratnaparki has had her lab at Agarkar Research Institute for more than a decade now. Anuradha did her PhD with Kaiti Hassan in NCBS and a postdoc in Caltech. And her lab is largely interested in looking at FGF receptors and how they signal in the Drosophila nervous system. But apart from that, they've also done this really beautiful work with a cytoplasmic protein, very conserved protein called MON1, that they've shown is required for maintaining postsynaptic receptors and is also required to maintain the gonad, especially the ovaries in Drosophila. The last lab that I'm going to be telling you a little bit about is Vatsala Thirumalai's lab. And she also has had her lab for about a decade at NCBS. And her lab works on zebrafish. And they do some beautiful work on looking at neuromodulation in zebrafish. And have recently shown the role of dopamine receptor in visuomotor behaviors in zebrafish. They also look at the expression of different different transcripts in the zebrafish brain. This is just a snippet of some of the labs that work on model organisms in India. And I think this is also a beginning if somebody wants to look at more labs. Dr. Shruti Muralidhar has made a very extensive list, and thank you for that, because I use that list to look up Indian PIs. Working in neuroscience in model systems. And with that, I want to thank you all for your attention. And I also want to thank Sandhya particularly, because she helped me a little with the talk in the initial draft of which she gave me her comments. Okay, and I'm going to go on to introduce the first speaker for today. The first speaker is Dr. Swagata Dey. Swagata completed her PhD from TIFR Mumbai with Krishanu Ray, where she did some really beautiful work on anterograde transport and how it regulates synapse organization in Drosophila neurons. She then went on to NBRC as a DST Inspire faculty and is currently a DBT Wellcome Trust India Alliance Early Career Fellow. She made the really smart move from Drosophila to C. elegans at this point. And she can get her slides up. She works, she's essentially looking at how cytoskeleton affects dendritic regeneration. And the title of her talk is Revisiting the Neuron Doctrine, Insights from Live Imaging. Thank you for that. Thank you all. And Swagata, over to you, take it away.
Thanks, Kavita, for that introduction. And I would like to thank the organizers for giving me this opportunity to showcase my research and career. But before I dive into my work, let me go down the memory lane for a bit. As a kid, I was really curious about various things, and I used to ask all these questions, and people around me had a hard time answering those questions. And I think everyone here can somewhat relate to it. We are all curious beings. And we have tons of questions that keeps our brain busy. And we think, and this tendency of thinking drives us to question and address what makes us who we are today. And today I'll try to explore some of these questions about myself and my work. Let's see. Let's start a timeline from 19th century. Like in 1873, when Camillo Golgi was trying to develop a staining mechanism to see cells, and including neurons. And a decade later, Cajal was interested in the same staining reaction, and he was doctor by profession, but he was an artist at heart. And he was fascinated by these stainings, and he used his skills to observe and depict neurons in his drawings. But what was special was his inferences. For example, here he described retina as a population of neurons that are juxtaposed to each other so as to maintain a unique directional flow of information. And because of his contribution, he shared the Nobel Prize in 1906 with Golgi. Technological advancement has happened since then, and now the neurons look a lot different than what Cajal depicted. I was born here, sometime here, like in the era of technological advancement. And my research career started in 2007 when I joined the integrated PhD program in NCBS at TIFR. Since then, it has been an arduous but a fulfilling journey for me. I got my PhD in 2018 from TIFR. And after that, I joined NBRC as a postdoctoral researcher, and I have been supported by Inspire Faculty Grant and India Alliance Early Career Fellowship. I'm still in my early stages of my career, but I still have a long way to go. But today I'll make a stop here and describe my work to NeuroFemIndia 2021. So when Kahal was observing neurons, he also came across certain developing ones, and this gave him the idea about dynamic polarization of the neurons. So the neurons start as an unpolarized cells, they give out processes, these processes grow and mature, they form the axons and the dendrites, and eventually these will form synapses to specific partners in the nervous system. And this keeps on happening from before birth until old age. And as we learn things, these neural circuits evolve over time, and it integrates inputs from outside world and may make some structural changes in itself. And as we grow in age, it would be more about retaining essential connections. And with age, the circuit expand and consolidate, keeping the essential functions only. And eventually they degenerate due to aging. With so many changes happening in the nervous system, neurons are constantly challenged by the demand and supply problem. How do neurons are able to meet the biological demands of the organism? And this question has been I was interested in this question, and I wanted to pursue this problem of demand and supply. And for that, I went to the Drosophila nervous system. So when Drosophila embryo is developing, the nervous system is undergoing a lot of structural changes, and individual neurons are undergoing phases of neuritogenesis and synaptogenesis. And during these phases, the action areas of the neurons require a constant supply of molecules and membrane. So how these molecules are being catered to the site of action? Specifically, I wanted to understand how the cargoes that are being generated in the cell body are transported all the way to the synapse during the situation of a demand. And over the years, many studies, and including my own, have found that this axonal transport, which has been carried out by motors, they have been able to transport cargoes that are generated in the cell body to the synapse and even in the dendrites. And they are the instrumental molecules that have been carrying out this transport, much like any city highway. But the only problem is the axons are not like any other highway. It's a crowded one. And in the axon, there are a variety of cargoes like organelles, structural proteins, soluble proteins, and exogenous factors, which are being transported by a handful of motors. And this brings in the complexity of how these motors are able to transport this diversity of cargoes in correct quantity when there is a demand. And this question interested me, but how do we understand that? So I went through various motors, and kinesin 2 was one of the motors that is known to transport a diversity of the cargoes. Cholineacetyl transferase is known as a soluble cargo of kinesin 2. But when I started, there wasn't any bona fide vesicular cargo of kinesin 2. So there were some candidates, but one needed to scrutinize. And when I was in Gary Banker's lab as an exchange student, and that time I looked for possible RAB and motor interactions. And one of the putative candidates, RAF4, showed a very strong interaction with kinesin 2. But I needed to test this in vivo in some physiological or developmental context. So I came back to India and I started looking at the transport of RAF4 in Drosophila axons of kinesin 2 mutants. Now, kinesin 2 is a heterotrimeric motor with 2 motor subunits and a non-motor accessory subunit. And in the loss of function of this particular mutant, I saw that anterograde traffic of RAF4 and the synaptic enrichment of RAF4 was drastically reduced, which clearly now indicates that, okay, kinesin 2 is required for the anterograde movement of RAF4. And so is cholineacetyl transferase, or CHA. It was known to bind to kinesin 2 and get transported towards the synapse. But now how kinesin 2 is able to manage the transport of both cholineacetyl transferase and RAF4 at the same time? Wouldn't their quantities be different at the time of the demand? Interestingly, what I found that the synaptic enrichment of CHA and RAF4 were anti-correlated. What I mean by that is that when we looked at a loss of function of RAF4 mutant, we found that those synapses had more CHA. And at the same time, we see that there is less RAF4 and more CHA, and their mutants also show a very drastically polar locomotory phenotype. Loss of cholinacetyltransferase caused paralysis, and loss of RAF4 caused increased motility. Further on, we also found that during synapse assembly, low transport of RAF4 and high transport of CHA is promoted by kinesin 2 to enable this synapse assembly. And this competition between CHA and RAF4 is supported by signaling molecules like PI3K and synaptic activity, specifically at a developmental time point when synaptic region in Drosophila central nervous system is showing a marked enhancement. For me, this opened an area of cytoskeletal biology and neuronal responses to developmental cues. But then it also brings me to question whether similar mechanisms can work in adult nervous system when they are remodeling. And this is what I'm pursuing in my postdoctoral work. In order to maintain connectivity within the network, a typical neuron establishes a polarized structure with multiple compartments like dendrites, axon, soma, and synapse. And while axonal transport is maintaining the logistics in the neurons, the neurites are undergoing constant change. The input processes of the neurons, that is the dendrites, they are instrumental in determining the function and behavior. They receive the inputs, they process it, and they integrate so that a valid neuronal response can be generated. These dendritic arbors are a characteristic feature of the type of the neurons and are very correlated to their function. Like for sensory neurons, the dendritic field is expanded so as to receive and integrate inputs efficiently. And this dendritic morphology is correlated to certain neurodevelopmental disorders and acute clinical conditions. So unlike axons, the dendrites undergo constant remodeling throughout the lifetime of the organism. A few of the examples I have listed here. For example, class 4 DNA neurons in Drosophila. In the larval stages, it has a very different morphology. And as it becomes the adult, the dendrites acquire a very different morphology, which is supported by the underlying substratum. It can also, the dendritic morphology can also be altered to refine the circuit as seen in mitral cells, which had connections with multiple glomeruli, but eventually after receiving a sensory input, they refine it to a single glomerulus to make an efficient connection. These dendrites can also remodel under certain acute pathological conditions, something like ischemia, where you can see these dendrites of the pyramidal neurons, they have actually remodeled quite a lot as compared to the control. So it seems that the dendrites are remodeling with respect to some environmental changes as well as certain intrinsic changes. And these intrinsic changes and extrinsic changes are able to maintain this dendrite remodeling while maintaining these essential connections and a coherence in the behavior. Inability to do so causes a degeneration in the neuronal compartments, loss of sensory or motor modalities, neuropathological symptoms. So now I became interested in that if this kind of remodeling is happening, the underlying cytoskeleton must be collaborating with that. So how does an underlying cytoskeleton is enabling this kind of remodeling? As dendrite arbors can be complex in structure, not many model system are available, but Kavita has already explained a lot about C. elegans, so thanks to that. C. elegans, because of their transparent body and relatively simple nervous system, they allow observation of neuronal anatomy in vivo, and especially during long remodeling events like dendrite development and regeneration. Now, PVD neurons in C. elegans, they provide an excellent platform to understand this problem. It's a neuron with a well-defined axon and a well-arborized dendrite. It has primary dendrites which are running in anterior-posterior direction, and it has orthogonally running branches giving out to secondaries that branch into tertiaries and quaternaries as they develop. And this hierarchy also is maintained by, also is visible in their cytoskeletal constitution. The primaries have a very microtubule rich cytoskeleton as compared to the secondaries, tertiaries, or quaternaries. On the other hand, the tertiaries and quaternaries have an actin-based cytoskeleton, unlike the primaries. So this gives me a system where I can actually study each of these neurites and how they are developing or they are remodeling with respect to certain changes. To study dendritic development, I have established a live imaging paradigm where distinct developmental stages can be imaged. The primaries are laid out by L3 stage of the C. elegans development. And following that, the other branches are laid out in a particular hierarchy. And as we can see that during the live imaging, we were able to see these dendrites getting formed, and also they are growing and retracting at the same time. But simultaneously, they are also assessing their territories for a consolidation or an avoidance purpose. And in order to facilitate this dynamics of branch formation, growth, and retraction, we speculated that it is correlated to the microtubule dynamics of this particular neuron. While we were looking at possible microtubule regulators, we came across a mutant, KLP7. But we found that this KLP7 had a very had a branch density which was very different than the wild type. And when we started quantifying these branch density, what we found that these secondary branches were drastically increased in this particular mutant, whereas the tertiary and quaternary were also comparably different than the wild type. Our live imaging also showed that there is a dynamic equilibrium in the branch density, which happens during the development. And this was highly perturbed in the KLP7 null mutant. So what is KLP7? Now, KLP7 is a motor of kinesin 13 family, and it is required for the depolymerization of the microtubules. It can associate with both plus ends as well as minus ends of the microtubules, and it depolymerizes the microtubules in an ATP-dependent manner. Originally, it was discovered during mitosis, where it is supposed to be localizing with the centrosomes, and it chews back the microtubules during the anaphase movement of the chromosomes. But it also regulates the anatomy of the touch neuron, and that has been observed in the ALM. And you can see that in the KLP7 null mutant, ALM shows a ectopic extension. It is right now, however, unclear how KLP7 has a role in dendritic morphogenesis. As KLP7 is known to affect the microtubule dynamics and orientation, we observe the dynamic microtubules by tracking EBP to GFP, which marks the plus ends. Firstly, in the wild type, we see the major dendrite has minus end out. That means the comets of EBP to GFP are moving towards the cell body, whereas the minor has a plus end out orientation of microtubules, very similar to axons. That means comets are moving away from the cell body. In the KLP7 mutant, we did not see a drastic difference in this microtubule orientation. In fact, it was very similar to what has been observed in the wild type, which suggests that KLP7 is not the sole regulator of microtubule orientation in these dendrites. What was interesting, however, was the localization of these dynamic microtubules. Now, in the wild type, the dynamic microtubules were restricted to only the primary majors and major and minor dendrites and the axon. But in the KLP7 null mutant, we were able to see dynamic microtubules that are invading the territory of secondary or sometimes even tertiary, which suggests that probably KLP7 is required to restrict the microtubules from invading the dendrites which are higher order dendrites, or which have an actin-based cytoskeleton. In order to find out more, we looked at the localization of KLP7 in these dendrites using a single copy insertion line. Endogenously expressed KLP7 GFP was localized to certain dendritic branch points, which can be seen here, that some of the branch points do show an increased levels of an enrichment of KLP7 GFP. And this enrichment was further enhanced when in the adult stages, when the more number of incipient branches are formed. So now we have more questions than answers actually. So it is possible that KLP7, just like in mitosis, that it associates with the centrosomes, and along with gamma tubulin, it helps in the retraction of the microtubules. Probably here also it is doing the same thing. And we would be actually investigating this further using live imaging of EBP2, KLP7, and gamma tubulin in the dynamic branch points to see how these proteins are actually collaborating together to maintain a particular branch density in a adult life. So what we think is happening here, that this growth and shrinkage of the branches is an alternative to classical dendritic pruning that has been observed till now. So in conventional dendritic pruning that has been observed till now, branches thin out, they get severed, and this is dependent on microtubule depolymerization and severing. What we think that PVT blueprint or PVT arbor blueprint is based on axonal trajectories of other neurons and spatial distribution of muscles and epidermis. And we think that this particular mechanisms where KLP7 mediated depolymerization or gamma tubulin based nucleation and EBP2 based polymerization, this kind of mechanism gives a calibrated approach to cover the territory instead of making the territory first and then reducing it. So this might be an alternative to dendritic pruning that we already know. We would be investigating other cytoskeletal reporters to approach this very question, and maybe I will share more insights later. Dendrites can also undergo extensive remodeling following injury. Now, Kahal, when he was exploring neuroanatomy, he came across injured and regenerating nerves. His primary observations here depict an unchecked growth of neurites in and around the injury area. Over the years, research on neuroregeneration has revealed that PNS neurons have a better regeneration capability than the CNS neurons. Still, in certain scenarios, the regeneration is aberrant and may cause deteriorating symptoms like paralysis, seizures, neurological damage, sensory and motor modalities are lost. However, unlike axon regeneration, dendrite regeneration is not understood well. Critical regulators for axon regeneration is known, something like DLK1, but few evidence indicate that dendrite regeneration is independent of DLK1 signaling. There is some cytoskeletal remodeling that is happening, but we don't know what is causing that. There is no molecular determinant for dendrite regeneration as of now. So in order to understand that, we use a PVT neuron again to observe the dendrite regeneration in this particular, in the worm. And what we found that we used a dendrite injury paradigm using a femtosecond laser. And after 24 hours of injury, we see a growth that is emanating from the severed end. And that is almost covering the same territory as the original dendritic arbor. And as time progresses, this arbor grows and tries to cover the original territory to its fuller extent. But we also see that during the process, it is doing fusion with other kind of branches. And also there are crossovers and there are formation of ectopic branches. But what is the cytoskeletal basis of it? We need to still understand. Now, in the axon regeneration, it was known that DLK1 was initiating the cytoskeletal remodeling in the first place. And later on, it is building the transcriptome that is allowing the cytoskeletal remodeling later to heal the injury. We did not know that whether DLK1 is required for the PVT neurons. So we looked at the axon injury in PVT. Axon injury in PVT also causes this axon to initiate a regeneration from the severed end. There is a regrowth, but we also see formation of ectopic branches and repurposing of the adjacent dendrites to serve as a function of axon. They show mislocalization of axonal cargos, suggesting that they are trying to serve the purpose of an axon. When we looked at the DLK1 null mutant, we found that the axon regeneration was perturbed in this particular mutant. And which suggests that, okay, just like other model systems, that DLK1 is required for the axon regeneration in PBT neurons. But what about the dendrite regeneration? When we looked at the same mutant for dendrite regeneration, we found that the dendrite regeneration in DLK1 null mutant was comparable to the wild type, which suggests that okay, DLK1 is not required for the dendrite regeneration in PBD neurons. But then what is exactly regulating the dendrite regeneration? Over many candidate regulators we screened, we found that SET10 is one of the mutant where the dendrite regeneration was perturbed. Now, SET10 is a GTPase, which is required, which is upstream of both microtubule and actin. So it is possible that when the dendrites are injured, the SET10 is locally changing the cytoskeleton so as to remodel the dendrites in some way. We would be looking at various actin and microtubule reporters to see how the cytoskeleton is responding at that particular event. But that would be a discussion for a later time. Okay, so my journey on neuroremodeling and its cellular basis has explored different neuronal compartments during their phases of remodeling. And the more I learn, the more I get inquisitive about the cellular nature of these events. I hope to share more insights on this in future. I would like to thank, I have been very fortunate to receive support from mentors and peers who have been instrumental in shaping me what I am today. My alma mater and current institutions have provided me the necessary infrastructural and academic support. The collaborators have given an alternative perspective to this research questions that I'm interested in. And of course, it would not have been possible without the funding. And thanks for your time, and I'll take some questions.
Thank you so much, Swagata. But first, thank you, Kavita, for this very nice overview which you gave on model systems and how they're useful to study neural, cellular, and molecular biology. And thank you, Swagata, for this wonderful talk. We have some time for questions, so I'll take up the questions which have come up in the Q&A box. Starting with the question from Vaishnavi, since it's at the top and has the maximum votes. So Vaishnavi's question is, does kinesin 2 have different adapters for RAP4 and chat, and how is their expression correlated?
Okay, so the previous work in Krishanu's lab before me, they found that CHA and kinesin 2 form a very small complex, and they are directly interacting with each other. And for my study also showed the FRET data, which shows that they are directly interacting. For RAP4, we know that it is not directly interacting, but there might be some adapter which Krishanu lab is pursuing right now. So there can be adapters like Rabaptin5 or Rabip4. So these adapters can associate kinesin 2 with RAP4 vesicles, but as of now, we don't know the adapters.
Okay, thank you. Next question we have from Ankush Chakrabarti. So first, he compliments you, really interesting talk. And his question is on, has there been NMR spectroscopy studies in adult humans to look at the chat and RAP4 expression with respect to brain structures?
There has been, so it is, people have looked at RAP4 and CHA in certain contexts, something like in people who have been, brains of cognitive deficient patients have been looked at, and it has been shown that high RAP4 was found in those specific brain areas. So some of the research has been done, but it has not been done extensively at the molecular scale. But overall, there is some expression level data that is present for both CHA and RAP4 in human brains as well as other model systems.
Okay. Abhilash, I would like to know if the axon restricted to only some portion of the body, or does it also span all the entire warm body?
The axon is, so this particular neuron, PVT neuron, is located mediolaterally. So the axon begins on the lateral side, it goes to the ventral and then moves in the anterior direction. And it does not reach all the way to the anterior, it stops somewhere after the midline. So it does not span the entire, but it fasciculates with the ventral nerve cord, which also runs throughout the length of the worm.
Okay, and here's one more question. Do the effects of the axonal and dendritic regeneration upon injury change the movement profile of the animal? Curious if different kinds of injuries like chemical or physical would have a similar kind of regeneration?
Okay, that's actually a good question. So we have looked at some of the behavior data, but so the behavior PVT neuron is responsible for is harsh touch. That means, and the posture. And it seems these parameters are slightly affected in neurons which have undergone exotomy or dendrotomy. And there are certain other studies who have also looked at the behavior, and in those also it seems the PVT neuron is required for these changes. Now the thing is chemical injuries and physical injuries. So right now we are doing a laser-based injury. So it definitely initiates some of the molecular changes that are required for an axon or a dendrite to sense the injury and initiate a response. I'm not sure about how the chemical injury would be done locally, but we have not actually checked the chemical injury patterns, but I'm assuming if they create the similar response, something like calcium transient and initiation of DLK in axon, then it might actually create a similar response.
Okay, great. Thank you so much, Swagata, for a wonderful talk. And we will now move on to our next speaker. We
Thanks.
have a very small slight change in the schedule, and Neha is going to be, Dr. Neha is going to be speaking next. So Neha, please feel free to set up your talk as I introduce you. Yeah. So Dr. Neha Jain is an assistant professor at the Department of Bioscience and Bioengineering at IIT Jodhpur, where she and her team work on understanding the formation of microbial amyloids and their role in biofilm infections. They also develop strategies to combat amyloid-derived biofilm formation by using a combination of biophysical techniques under different environmental conditions in vitro studies. She's a recipient of the 2019 ASM IUSSTF Visiting Research Professorship and also won the 2019 Early Career Research Award from DST SERB. Today, she's representing the neuronal diseases field and will talk about how bacterial amyloids influence human amyloids to try and understand the role of gut microbiota in the onset of Parkinson's disease. Dr. Neha, thank you so much for accepting our invitation to talk in this conference. Over to you now.
Thank you, Bhavna. Am I audible and is my screen
Yes,
visible?
yes, your slides are also seen. You just have to make them in full screen mode.
Yeah, that's what I'm trying to do.
Yeah.
Is it full screen visible now? No?
Not yet.
Okay. Yeah, is it okay now?
Yes, perfect.
Yeah. I'm really thankful to Bias Watch for giving me this opportunity. As well as I would like to mention that I'm not a neurobiologist, so I'm really sorry for disappointing some of you. I'm a protein biochemist and biophysicist by training. We try to understand protein folding and misfolding that contributes to Parkinson's disease. So hopefully by the end of this talk, I would be able to convince you that the work that we do is related to neurobiology and is would be able to answer some of the fascinating questions in the field of neurodegenerative diseases. So I have recently joined the Department of Bioscience and Bioengineering at IIT Jodhpur in 2019. So I'm a young faculty here at in terms of joining. And I have a lab here, which is called as Functional Amyloid Biology Lab, because we work mostly on the amyloid perspective. So as most of us know that there's a lot goes behind a normal brain to get converted into a diseased brain. And there are many factors that contribute to this conversion over a period of time. So what are these factors that lead to this conversion? There are multiple factors that include mitochondrial dysfunction, ROS generation, neurotoxic oligomer formation, metal ions, genetic factors. All these factors, either individually or in combination, lead to formation of these uh, aberrations in the brain and causes neurodegenerative diseases. Among all these factors, one of the major factors that contributes to this neurodegenerative disease in most patients is deposition of amyloid fibrils. And today, my talk will be mostly focused on amyloid fibrils and how these fibrils contribute to neurodegenerative disease. And here I will be talking not just about one fibril, but I will be talking about two different fibrils. And if you look under microscope, these fibrils look like these rod-like structures. So in my couple of slides later on, you will see a lot of images like this, which will have thread-like structures. And these are actually amyloids, which get deposited in the human brain. So before I start my talk, I would like to give you a brief introduction on what are amyloids. These amyloids are the protein structures which were initially identified as carbohydrates by Rudolf Virchow as back as 1854. So these structures were stained by uh, staining dyes which were used to stain starch, and that's why Rudolf Virchow called them as amyloid or amylo. Later on in 1958, Friedrich and Kekule actually identified that these structures were not carbohydrate, rather they were proteins. And that's where the field got changed and people got interested more that there is something going wrong within the proteins and which is causing these diseases. So let's look into more details about these amyloids. So amyloids are basically the change in the conformation of proteins. So usually we have a protein which is natively folded, which is performing its function. However, sometimes this protein doesn't go into its native conformation, which leads to misfolding. Most of the time in our body, it is being taken care by chaperones or chaperone-like proteins. So whenever the protein misfolds, it goes back into the protein degradation machinery and it gets degraded. However, as we age or under certain environmental conditions, these misfolded proteins keep on accumulating inside our body. That leads to formation of the amyloids. And to not to great surprise, this amyloid causes a lot of diseases like Alzheimer's, Parkinson's, Huntington's. So for many, many decades, it was considered that amyloids are the flaw in the system. That means the protein misfolds and it leads to amyloid formation. However, later in 2002, Matt Chapman from University of Michigan identified that these amyloids actually play an important role in different aspects of life. So he figured out that bacteria makes these amyloids on purpose, and it utilizes this amyloid fold to protect itself. And now we can see that there are many amyloids which have their function, and it is present in all forms of life, starting from bacteria, yeast and fungi, and animals. Among these functional amyloids, curli or the bacterial amyloid is the most well-studied amyloid form. Curli is present in Escherichia coli, where it forms a network on the cell surface. So if you can see, this is an E. coli cell, which forms a dense network of these amyloid fibrils. These amyloid fibrils contribute to biofilm formation. And so now we have two categories of amyloids. One is disease related and one which has function. No matter whether they are disease related or they are functional, they are formed through a similar pattern. So basically, your protein undergoes a conformation change, they form oligomers, and they finally connect together to form these fibril-like structures. Now, why these fibril-like structures are so important? Because the formation of these fibrils is a generic property of all the proteins. That means any protein at a particular given condition can be transformed into amyloids. They are composed of highly unbranched cross beta sheets That means they are just intertwined like this to form a very robust structure. With just one exception, which appeared last year in Nature, where a protein from Staphylococcus aureus was found to be made up of alpha helical structure. The amyloids were made up of alpha helix. Other than that, all the amyloids, without any exception, are made up of these cross beta sheet structures, which give them highly stable structure. These become non-degradable and they are resistant to any kind of denaturation. So now we can think of that if these amyloids can start depositing in our body, there is no mechanism by which we can degrade them. Now, it's very important to understand this amyloid process under the lab conditions so that we can create certain remedies to overcome the formation. So, this amyloid assembly can be understood well in the lab conditions by using a proxy for amyloid formation. So, basically, what you do is you take a monomeric protein and you monitor the change in the conformation as a function of time. You use a fluorescent dye that can bind to these fibrils. So generally, half-level is the fluorescent dye that we use commonly in the lab to monitor this fibril formation. Most of the time, the amyloid assembly follows three steps. The first is the lag phase. I'm not sure if my white pointer is, let me just take it there. So first step is the lag phase, where the monomeric proteins, they assemble together to form a critical mass, which that gets converted into oligomers, which finally starts making the fibrils and the protofibrils, which is represented by the elongation phase. And then when the almost all the monomers are converted into fibrils, which is represented by the stationary phase. So this is a kind of a curve which you are going to see in my slides many, many times. So I would like you to pay a little bit more focus on this, that we have a lag phase, we have an exponential phase and a stationary phase. This is a general amyloid assembly pattern. Now, if you have a decreased lag phase in any protein by adding any molecule, that suggests that that particular molecule is accelerating that amyloid formation. If you get a flat line after adding a certain molecule, that means that molecule is inhibiting the amyloid formation. So that's why this graph becomes very important to understand whether certain biomolecules are accelerators or inhibitors for amyloid assembly, and can we use those biomolecules for therapeutic interventions. So let's start with the talk finally now. So Parkinson's disease, as we all know, that it's a neurodegenerative disease. So under the normal conditions, this alpha-synuclein protein, it is localized at the synaptic terminal. Although the exact function of the synuclein protein is not well understood, however, it is known that it is required to maintain the neuronal plasticity of the cells. So under normal conditions, you will have dopaminergic neurons which functions very nicely. However, when under certain circumstances, maybe you have mutations, maybe you have certain environmental factors, so where this alpha-synuclein undergoes conformational change and form oligomers that gets converted into protofibrils and fibrils. So during this process, what happens to the cell is that due to accumulation of these oligomers and fibrils, the cells start to die because they are not getting the required amount of nutrient and the oxygen that is required. This cell death leads to the movement disorder. Now, there are many scientists all around the world who are trying to understand that how the aggregation of alpha-synuclein triggers Parkinson's disease, and what are the different factors that contribute to this progression of this PD. But there is no one conclusion. So there are many concepts which says that this could be the right one, that could be the right one. However, we don't know exactly when alpha-synuclein actually starts making fibrils. So it's very difficult to detect the early oligomers or the protofibrils in a Parkinson's disease patient. By the time the disease is diagnosed, it's already too late. So now what we want to understand is that whether there is a crosstalk between bacterial amyloids and human amyloids. So whether the bacterial amyloids have any effect on human amyloids. Now, synuclein aggregation is itself very complicated and a lot of scientists are still working on it. So why do we want to create one more complexity to this already complex phenomena? Why we want to venture into this? Because there are many reports which suggest that there is some kind of connection between Parkinson's disease and gut microbiome. These connections are indirect, which shows that the gut microbe has something to do with the onset of the Parkinson's disease. It is still not clear that which portion or which metabolite, which byproduct of these microbes which are present in the gut are responsible for either the progression or in the continuation of Parkinson's disease. So there may be many different ways to answer this question, that how do we figure out that there is a connection between Parkinson's and gut microbiome? It could be secondary metabolites, it could be cell walls, the cell membrane components, or any other byproducts. But we took the approach of amyloids. The only reason that we took the approach of this amyloids, because I had a previous expertise of amyloids. I did my PhD in amyloids. So we thought that let's see whether alpha-synuclein can crosstalk with a microbial amyloid, E. coli. And if this crosstalk happens at the protein level, and whether this crosstalk has any significance in the progression of Parkinson's disease. So here I would like you to just remember that we're dealing with two proteins. One is the microbial protein, E. coli, and we are purifying protein from it, which is amyloidogenic, and we are dealing with alpha-synuclein, which is again making amyloids. So these two proteins are evolutionary very, very apart. And usually the evolutionary apart proteins, they don't interact with each other. So we didn't initially anticipated that there will be any crosstalk between these two proteins, but surprisingly there was something. So how did we started this study? So let me just first tell you how these two proteins individually form amyloids. So to understand this, we follow a very basic study in our lab. We study protein-protein interaction by using different techniques like fluorescence spectroscopy, some biochemical techniques, microscopy techniques, and cell-based cultures. So by using these techniques, we understand protein-protein interaction, and we want to figure out that can we understand the molecular mechanism of amyloidogenesis in more details? Can we figure out the role of microbial amyloids in neurodegenerative disease? And can we find out the anti-amyloid therapy by using this kind of strategy? So before going into detail, I would just like to show you that alpha-synuclein is a human protein which is not supposed to make amyloids, right? It makes amyloids only under certain conditions. So that's why a very high protein concentration is required, and you need certain agitation, and it takes long time to make amyloids. However, coli from E. coli is meant to be an amyloid. That's why if you just throw this protein on your bench, it will just aggregate by itself. It will make amyloids within a couple of hours. And you need a very low concentration of this protein to make amyloids. Okay, so how did we start our studies? We first monitored the synuclein aggregation alone. So it typically follows a nucleation dependent phenomena where we have a lag phase followed by exponential and stationary phase. Now, at the same time, what we added is that we took synuclein in a next set of the experiment, and we added purified soluble CSG. That means this is a bacterial amyloidogenic protein. We added to this synuclein, and what we found is that when we added this protein, we see a change in the kinetics of synuclein aggregation. Now, the point here to be noted is that this CSG that we are using here is a very low quantity. So you can see that the ratio is 1:25. Synuclein is much excess as compared to our CSGA. When we looked deeper into this graph, we found that the lag phase for synuclein was around 18 hours. So this time period was around 18 hours, whereas with CSG, the lag phase dropped almost to half. And the rate of the reaction was also different. So we figured out that CSG is accelerating alpha-synuclein amyloid assembly, and it's also modulating the rate of the reaction. The second question was that whether CSG is bringing any change in the secondary structure of the protein, because the protein is now getting transformed into highly beta-shaped structure. So next we monitor the change in the secondary structure. So at 0 hour, both the proteins are in the random coil because both proteins are intrinsically disordered proteins, so they do not have any structure of their own. After around 13 hours, what we observe is that the sample of synuclein, which was with CSGA, undergoes conformation change and starts forming beta sheets at a much earlier stage as compared to the sample with just synuclein. After 60 hours, we saw that both the samples got changed into beta sheet. However, here we do not see any change in the content of the beta sheet. So we analyzed this data to figure out whether the beta sheet content is different, but we found that the beta sheet content was not different. So the next question that we asked was whether there is a change in the morphology of these two fibers when you have CSGA and when you don't have CSGA. So we looked these samples under transmission electron microscope, and at the zero time, we, as expected, we didn't see any fibril formation in any of the samples. However, after 60 hours, we found that the fibers that were formed were grossly same in presence of CSGA as well as in absence of CSGA. So this is just a human protein. This is human plus bacterial protein. And thing to be noted here is that just the CSGA protein didn't form fibrils by itself. So here we didn't see much of a difference. However, I should confess that we didn't go into the details of this fibril morphology as to figure out minute differences like whether the branching pattern was different or the twist was different. So this is something that we are going to carry forward later on once the situation becomes better. So in this condition, however, the gross morphology was found to be same. So now we know that CSGA, which is a bacterial protein, it accelerates synuclein, but the overall morphology, the overall change remains the same. Now, when we were doing these experiments, so the data I've shown you, I've done this in my postdoc lab at Matt Chapman at University of Michigan. So when we were doing this in Matt's lab, another lab at Caltech, which is Sarkis Mazamian's lab, they were also interested in looking at how E. coli, that is expressing curli, is affecting Parkinson's. So we talked to each other and we thought that let's club these data together to have a more robust story. So I'm just showing part of it to show that our in vitro data actually corroborated well with the in vivo data. So what Sarkis lab did is that they took the wild type E. coli and the CSG-BAC mutant from Matt's lab, and they inoculated these strains into Parkinson's mice, which is germ-free mice. So the mice were either wild type or alpha-synuclein overexpressing mice. So if you see here, when they were inoculated with wild type E. coli, which has a functional curli production, the alpha-synuclein overexpressing mice has an increased time of descendant in the pole test. So pole test, I hope you all know, but the pole test is nothing but you take a pole and you check the time that when the mice, how much time the mice is taking to descend down. So a normal mice will not take much time. However, mice with any neurodegenerative disease will take a longer time. So this shows that the alpha-synuclein overexpressing mice with E. coli producing curli took longer time. However, with the E. coli that could not produce curli, the results were indifferent. This test was further confirmed by sticky note test. The sticky note test is again a very easy test, which what you do is you put a sticky note on the nose of the mice and you see that how quickly that mice is able to remove that sticky nose. And we got the similar results with this, that the alpha-synuclein overexpressing mice with the wild type E. coli took longer time to remove this. So this suggested that whatever we were looking at in vitro, that CSG is having impact on alpha-synuclein, we were able to see the similar effects in the in vivo studies. And then Sarkis lab also did a lot of immunological experiments to prove that there is some connection between CSGA and curli. So this is where when I switched my career and I moved back to India at IIT in 2019. And then I thought that since I'm a biochemist, let me proceed with this study and let's figure out that what is there in these two proteins that they are interacting with each other. So there is a very common phenomena in amyloid field, which is called as seeding. That means you take these fibrils, you break them apart, so the ends become very sticky. And when the monomer comes to it, they again go back into fibril formation. So that is called as a seeding phenomena. And if you take synuclein seeds, the monomers quickly go into the fibrils. So there is no lag phase. So I thought that CSG could be acting as seeds for alpha-synuclein. So I took CSG seeds, but didn't see any change. So that means it's not a seeding phenomena. Rather, the CSG monomer was having some sort of interaction with alpha-synuclein, and that interaction is accelerating the aggregation of alpha-synuclein. The next question then I asked is that whether there is any sequence specificity to this interaction. So next we looked into client specificity. So what we looked into is two other proteins, tau K18 and amyloid beta. These two proteins are also involved in Parkinson's as well as Alzheimer's disease. So we saw that CSG had no effect on either tau or A beta, which I'm not showing here right now. So this suggests that CSG has client specificity, that it's not accelerating any random protein. So there was some specificity to its activity. So where that specificity was lying, to understand that, I dissected the sequence of CSGA. So CSG has 5 regions, which Maths Lab has beautifully described in earlier papers. There are 2 important regions, R1 and R5, which are critical for aggregation of CSGA. So what we did is we removed these 2 regions and we then did the same experiment. So we found that if you remove R1 and R5, there is the effect, acceleration effect is gone. So we thought that this is very drastic effect. Let's remove one region at a time. So then we removed R5 first. We saw that again the effect is gone. When we removed R1, again the effect was totally gone. So this suggests that R1 and R5 might be important in interacting with alpha-synuclein. However, the question was also that if you're removing such big regions, just that protein might not be behaving normally. So we decided to remove a region which was not important in CSG aggregation. So what we did is we removed the R3 region, which was not important. And what we found is that if we remove R3 region, the acceleration activity was restored. So this suggests that the aggregation competent conformation of CSG is required for acceleration. Next, we looked into the alpha-synuclein C terminus for the interaction. So the C terminus of alpha-synuclein is usually involved in interaction with other proteins and ions. This is a region which maintains the intrinsically disordered state of the protein as well. So if you remove the C terminal, the protein will aggregate pretty much faster. So when we deleted this C-terminal region, we found that, of course, the lag phase was reduced and the effect of CSGA was also diminished. Now, as of now, we don't know whether it diminished or since the lag phase is already very short, CSGA didn't have any impact on that lag phase. So this is still, we need to figure it out. So we think that CSGA might be involved in this kind of study or not. Now, in a couple of days back, we heard Dr. Indrani Datta, and she mentioned that the wild-type synuclein and the PD-related mutants, they behave quite differently. Okay? So whatever studies we have done so far were on wild-type alpha-synuclein. So we then did the similar studies on Parkinson's-related mutants. There are around 7 PD-related mutants. However, we have just tested only 3 of them right now. So one is A30P, which didn't show any change in the progress. Then we used A53T, which showed the similar effect as wild type, and A46K, which didn't show. So this also suggests that the CSGA effect on the aggregation of alpha-synuclein might also be dependent on the mutations that are already present. So what we conclude from our studies is that a bacterial protein which has a capability of making amyloid can alter the human aggregation. The fibers, they have the similar gross morphology with same biophysical and biochemical properties. There are specific regions in both CSGA and alpha-synuclein which we think are important for this crosstalk, and this crosstalk is specific to certain mutants in the Parkinson's disease. Now the question is that how does it matter in the area of neuroscience or neurobiology or neurodegenerative disease? So what we think is that it's not just the protein which is aggregating, which is alpha-synuclein which is aggregating. However, there could be many other proteins from the microbes that are aggregating and that might be contributing, and we may have to specifically tear them apart to figure out that one Parkinson's patient might have different symptoms and different progression as compared to other, because PD patients do have different microbiome as compared to the normal patients. So what we think is that the microbial amyloids play a very important role in neurodegenerative disease. And what we need to look into is that what are the different environmental factors that contribute to change in the microbiota of human beings that might lead to become microbes as a cause or effect for PD. How does the immune response to these heterofibers would takes place? So during my ASM fellowship, I actually looked into the immune response of these heterodimers, and we found different levels of interleukins with just alpha-synuclein and alpha-synuclein plus CSGA. So if we can detect these immunological differences between the patients, we might be able to detect the disease much earlier, at much earlier stage. So with this, I would like to thank Matt Chapman's lab at UMich, where I started this study, and my current lab here at IIT Jodhpur, along with different funding agencies, which allowed me to start my work here. And with this, I would like to thank all of you for listening to this talk, which was not very much into neurobiology, but thank you so much for your time. And thanks, Bhavna.
Thank you so much. Yes, thank you so much, Neha, for your very nice talk on Parkinson's and the gut-brain connection. I'm sorry, our next speaker, Dr. Uttama, will have to leave by 3. So, in the interest of time, I'll probably can take only one question live, and the rest, if you are okay with it, Neha, could you please answer them on the chat, or maybe we could come back to it later if you have time. Yeah, sure. Sorry about this, but I'll take the question which is at the top, which is from Poonam. And the question is, do we know how the microbial amyloids be entering the brains? Are they using the gut-brain axis, and do they interact with synuclein in periphery or CNS?
Yeah, a very nice question, Poonam. As of now, we do not know. But what we think is that synuclein is not just present in the brain, it's also present in the blood. And the gut epithelial cells, they also have a lot of synuclein. So we think that the transfer happens there in near around the gut. So the synuclein in the gut might get primed with the microbes, with these bacterial amyloids. And then when synuclein is circulating in the brain, then the other synuclein might also get primed, and it might lead to the aggregation of more alpha-synuclein. However, we do not have any direct answer to that. Sarkis lab is working on this, figuring it out.
Okay. Thank you so much, Neha, and please answer your questions from the Q&A box. Yeah? Thank you so much.
Thank you.
Yeah. Our next speaker is Dr. Uttama. Dr. Uttama, please, you could use this time to set up, and I'll introduce you. Yeah. Dr. Uttama Lahiri is an associate professor at IIT Gandhinagar, where she and her team work on stroke rehabilitation using interactive VR game-based tasks and developing low-cost sensors for diagnosis and measurement of peripheral physiology through virtual reality-based applications, modeling, and adaptive intelligent techniques. In 2019, she was given an award and chosen as one of 100 most dedicated professors, and in 2018 as one of top 50 women in education, both times by the World Education Congress. Today, she is representing the cognitive neuroscience field and will give a talk on computer-assisted diagnosis and rehabilitation of neurological disorders that employ virtual reality-based applications to address static and dynamic balance deficits of post-stroke patients. Dr. Uttama, thank you so much for accepting our invitation, and over to you.
Thank you, Bhavna. Very good afternoon to all of the attendees and the other panelists even. First of all, I would like to say that I'm not a biologist. In the way Dr. Neha Jain was also telling, I'm not a biologist. And in fact, I am a core electrical electronics instrumentation engineer who have been working at the crossroads of rehab and also on the cognitive side as well. So today my talk will be on computational neuroscience's role in post-stroke balance diagnosis and rehabilitation. Let me go over. Okay, so when I tell about balance rehabilitation, I refer to stroke a bit on the introduction. Why stroke? What is it? What effect does it have on balance, and how can we diagnose it, and how can we rehabilitate it? This is what my present day talk will be. today's talk will be. So it's actually a clot in the brain or a rupture of the blood vessel, which can lead to a stroke, and it's a neurological attack. And it is one of the most common causes of death and also disability, even in a global picture globally and also in India. And also it changes with age. The probability of the prevalence rate changes with age. More elderly people get more affected. But the worst side of it is even after they come out come out to the chronic stage, that is, come out of the very severe stage, they suffer from various disabilities. In fact, they have vision problems, they have balance problems, they have gait problems. Today, I'll be covering mostly the static and the dynamic balance aspects. Now we know that rehabilitation is necessary, and of course, earlier the rehabilitation, the better it is. But the problem is that in India, with a low doctor-to-patient ratio and the healthcare costs being unaffordable to many of the country people, we need a cost-effective, easily accessible solution, and which can provide quantitative estimates. This is something very important, because you will find the conventional systems are very subjective in nature, which have their own limitations. Now, in fact, you can see that in India, about 85% of the cases happen in the low and middle income countries, where India is one of them. And it is in India almost one of the top 3 causes of death. And also you can see that it's like nearly 10%. So it's like quite severe problem in India. Not only this, in the global scenario, there are about 15 million occurrences of stroke cases every year. And also out of them, 5 million nearly die. So it's like about 33% die. And out of those who survive, nearly 85% of those people have issues with gait and balance. And so this makes diagnosis and rehabilitation very crucial. So in fact, when the stroke happens, either it affects both sides of the body or it affects one side, that is paraplegia or hemiplegia. And this adversely affects their locomotion. In fact, they become dependent on others. And in fact, they are mostly wheelchair bound, which affects their community participation. So what the patients do, they go to the clinician, and in the conventional techniques, what the clinician does, they first assess the residual abilities. This is something very important for diagnosis. They assess the residual abilities and then accordingly prescribe rehabilitation exercises. And of course, based on the residual ability, they are offered rehabilitation phase of exercise. And in between, again, they are reassessed, and this entire process goes on until and unless their ability is improved, the post-stroke patient's ability is improved. But what I have told earlier, that the conventional techniques, though very powerful, are problematic, particularly in countries like India, given the low doctor-to-patient ratio. And you will find that these conventional techniques are very sort of subjective in nature, not objective. So how the conventional techniques in gait are actually practiced. So there are different tests. There are 6-minute tests, 10-meter walk tests. These are very well-known tests which clinicians use. And for that, these are like these are more stopwatch based, very traditional. And people generally mark their bottom of the feet with ink, and then they're asked to walk on a piece of paper about 10 meters long, and their foot imprints are being taken, and from there different gait parameters are being measured very manually. And of course, you can realize that this has a lot of issues. So obviously people are working on technology assisted solutions. So in fact, there are accelerometer-based systems like inertial measurement unit-based systems, which are very powerful, but as you all know, that these suffer from drift issues. So there are problems in repeatability. Again, there are motion capture-based systems like the Vicon. These are stereo photogrammetric systems, which are very powerful, very accurate, but they are mostly restricted to labs. and they are very costly. Also, not many labs can have them. Also, there are pressure sensitive walkways like gate ride. These are very precise, very good measuring methods. But again, these are mostly restricted to lab-based settings and they are very costly. Not many people can afford it. So people have been going for relatively portable solutions like pressure sensitive insoles where the pressure profile of the entire foot bottom is being recorded. But again, these are very costly solutions. So we sort of set out to quantify one's gait by bringing in instrumented insoles. So we made an instrumented insole which have different pressure sensors, and this is one of the one of our initial versions had 3 sensors. Now we have increased the number of sensors, and these are very portable solutions, and this can pick up the pressure points of the foot, and they can pick up certain gait events like heel strike events, toe off events, from where we can find out a host of different gait parameters. And we have designed interfaces which can offer a real-time estimate of how the toe sensor output is giving, how the heel sensor is output giving, and accordingly we can find out the gait events. So first we started out with, okay, since we have an instrumented insole, how good it is. We started calibrating it with the very standard stereophotogrammetric technique, which is the Vicon. And you can see we sort of asked the participant to walk on a 7-meter straight pathway, and intermediate like 4 meters, we sort of monitored both the Vicon and the shoe data. And from there, we carried out comparative studies to see how good our instrumented insole is with the standard techniques. We did with number of participants, nearly 15 participants were there to do the study. Now, additionally, this Vicon is very good. This camera-based techniques are very good, where you provide knuckles on the body and you observe under the camera. But the problem is that these mostly offer direct measurement of all the temporal parameters, not the spatial parameters, and not always the spatial parameters unless and until you have the speed information. So we tried to replicate the conventional paper-based, ink-based setup as well with our instrumented insole, where we went to the hospital, we marked the bottom of the feet of the shoe with the insole, with the ink, and asked the participants to walk on the white paper. And from there, we tried to see how our data matched closely, or what was the deviation with respect to the standard techniques. We also carried out with different healthy and also post-stroke. So 9 post-stroke patients were there in the study, all recruited from civil hospital. These were the different parameters, like almost 11 parameters we extracted from our shoe. For example, the step time, stride time. So step time, you can see on the figure on the right, the red shoe is the right leg and the left shoe is the left leg. The blue shoe is the left leg and the red one shows the right leg. So right leg to right leg, that is ipsilateral leg, is the stride, and the right to left or left to right, that is contralateral leg, heel strides, are the step. So we could find out the times relevant to step and stride, also the length information we were finding out. Also, we were getting number of steps per unit time, that is the cadence, which is something very important in different neurological disorders we see. We found out the walking speed. We also computed the stance and the swing phases, which are very important. People who are in the gait research, they know very well that so the stance phase is the amount of time where the leg is in contact with the ground, and the opposite. So one leg is in contact with the ground and other leg will be in the air, so sort of in the swing phase. And so you can get in fact the single support time, how much one leg is bearing your body weight. Also, we found out different parameters like walk ratio, gait stability ratio, which are again giving indicators of one's prevention towards falls, prevention for falls. So these are all gait stability parameters we computed. So the results indicate for the healthy participants about maximum of 1% was nearly about 1% for the stride, and the step time was the percentage error, the deviation. absolute error. So it was quite accurate to the very standard camera-based technique. For the healthy participants, as for the stride length and step length for the paper-based setup, also we could find out about nearly 1% at max was the error. And for the post-stroke patients, also we tried out and we could see about 1.5% was the error. So our shoe, our instrumented insole was working very well. A quick video, I felt, I'll skip the slides. I hope you can see it. So I'll just skip portions. So this is the smart insole, shoe insole. So this is how the insole looks. Impregnated in the shoe can mean how the gait is being quantified, the manner in which one walks. So this came up as a part of a real research project that ran over a period of nearly 5 years process. We went through validation, where we validated both across healthy and also against abnormal gait patterns. This shoe can record various spatiotemporal gait parameters such as stride time, step time. Even it can find out the stability ratio, which can prevent falls, which is an indicator of falls, and also the symmetry, the way in which we are using both the legs. Install helps in recording data in an autonomous manner and analyzing the same with a computer program in the course of treatment.
This wireless connectivity to get rid of all the wires hanging around.
The established wireless connection is stable enough over a long time and also accurately communicating over a long range. Dat je de zomerbanden ermee gelikt hebt en dan door het sneeuwstorm. Ja, dat zei je. We analyze various configurations of products so that it becomes visible without. Ja, het voordeel is inderdaad, het is niet een altijd sneeuw. The texture, color, and cushioning were picked in such a way so that it improves the functionality of it. The install GUI is an interface for user to visualize and record the show data. Later, it calculates gait indices from the recorded images. So the gait-related indices can be monitored and also recorded in computers, in laptops, wirelessly. I think so we stop at that. So you can see that we work through the entire life cycle. So sort of doing for the very basic studies, it was a bigger part of a bigger research project. And we go all the way to the design aspects. And also now we are getting interest being expressed by commercial partners. So, okay, now the gait has been quantified. It has been diagnosed. How do we rehabilitate gait disorders? So we went, that takes me to the next portion of the talk. In fact, you can find that the conventional gait techniques, gait rehabilitation techniques, they use very very good, but very subjective sort of techniques they use, sometimes a parallel bar, sometimes an orthosis for foot drop cases. These are, though good, these are very labor intensive. You can see multiple physiotherapists assisting a particular patient. And of course, these are quite costly, both direct and indirect costs. Of course, subjectivity is there. And of course, they are monotonous because it's very repetitive exercise. Now that brings us to the need for technology-assisted gait rehabilitation systems. You can see that there are different, literature shows that there are different robot-assisted systems, VR-based systems, physiology-based systems, and these systems show the importance of monitoring involuntary measures such as physiology, and also giving them a virtual reality-based interface, and also an automated system. So this is our system which we came up with. We have a treadmill-assisted training platform where we are monitoring one's gait using the instrumented insole, and we are offering a virtual reality-based interface, and there is an autonomous AI-based engine which will automatically and adaptively respond based on one's ability. So we mostly targeted 3 different points: physiology, sensitive, adaptive, and progressive. As you know, we were trying to tap the involuntary measures so that no one can control it. And physiology is one of the best involuntary measures. And it shows that if you make an exercise physiology sensitive, it can improve one's gait.
Of course, we wanted to make our system adaptive to individualized abilities. And so we wanted an adaptive system. And another new thing is we wanted to give a progressive training environment where we can increase the challenge level based on one's capability, where research shows that if you can increase challenge, then the person is more challenged and the rehabilitation improves. So we started with first comparing individual blocks. We went, so okay, we have a VR-based system and we have a same system without the VR, where we don't make it adaptive, but only have continuously increasing treadmill speed, where there is no adaptive like the one which is already available commercially now. So we prepared different types of virtual reality environments. For the interest of time, I'm not going to the details. And we also designed an optocoupler-based interface module where we were connecting the treadmill to the virtual reality-based environment. And we started with comparing VR against non-VR based tasks. Similar conditions, we kept same things. Both did not have adaptive, right? Both did not have adaptive, but one with VR, one with no VR. And we exposed almost 14 participants to an experimental protocol where there was rest, and also they were allowed to walk, sometimes in presence of VR, sometimes not in presence of VR. And we could see that in each case, there was an increased stride time. There was an increase in the stride time of VR compared to the non-VR. That means they were taking confident steps, much more confident steps, both for the left and the right legs. Also, the stride length also improved, again, showing that they are taking confident, bigger steps. Of course, the gait performance has improved. We could make out, but we could see that the VR based was giving better improvement compared to the non-VR based setup. We also saw the walk ratio, and we could see that also the walk ratio improved, indicating the spatiotemporal gait coordination between both sides of the leg. That also improved for the post-stroke patients. So then we felt, okay, let us bring in adaptivity in our system. So we could see that the stroke patients generally become exhausted very fast. Why? Because they spend more energy in doing an exercise. So how about we tap one's energy and one's energy consumption, and what are the physiological modalities by which we can pick up one's energy? In fact, we could see that the literature has indicated that if you increase one's walk speed, you can challenge. So our thought was, how about we pick up one's energy consumption during walking on a treadmill and then increase the challenge by increasing the treadmill speed. So that was sort of the idea behind. And then we thought that, okay, how about we find out energy consumption? Literature shows there are two different modalities. One is how much oxygen you consume. Of course, you can see on the figure that's a very cumbersome setup, and it's most restricted to lab-based settings. And the other one is a heart rate. So you can use a watch, you can use a very good portable data acquisition system, and you can pick up one's heart rate. And also one's heart rate is being very closely monitored during post-stroke rehab exercises. Of course, there are many parameters like submaximal exercise thresholds. If you are interested, we can speak later in details on these. So we wanted to make the heart rate adaptive closed loop system. These were the modules. I will go ahead with describing in briefly each of the modules. So we started with the VR-based task presentation. We had an avatar, and we were monitoring individual steps, and we gave an virtual environment, and the avatar speed was synced with the treadmill speed by using a VR treadmill synchronizer, the optocouple-based setup that we have used, that we have designed in our lab. Additionally, we made an adaptive engine, which is the AI engine. I'll not go into the details of this engine, which gives different checks and balances before telling whether we will increase the challenge or not. Of course, we see the thresholds based on one's heart rate as per literature, whether the person is taking any beta blocker medicine to bring down the heart rate, which is commonly used for post-stroke patients. We see all different variations. And then we gave the participants a treadmill-based walk. Of course, we gave a warm-up session followed by a rest, and then a treadmill-based session and followed by overground walk. And we did this almost for 2 months at a stretch for 9 participants. We monitored different parameters, but here we'll be presenting few of them. So this was the view. The patient was walking in Civil Hospital. The patient was walking on the treadmill. Of course, there was a safety harness. The person was wearing the shoes, and there was a VR display which was projected onto the screen, and there was a data acquisition system to collect the data. So we could see that the walking speed post training on the overground, this is the overground speed, the overground speeds improved statistically. So they, in fact, as per literature, they went to the community walking level. So really there was a big improvement. And mind you, this big improvement was at the expense of reduced heart rate. So that means they were literally saving on their energy consumption. So the PCI reduced. Not only their step length improved, and that too statistically, that is, they were taking confident steps. The step length improved, and also their gait asymmetry reduced. So you can see the red bars are for the affected leg, the unaffected leg. So you can see the red bars are affected leg, and the green bars are for the unaffected leg. So you can see, and in the first case for the affected side, the percentage during the pre, there was a statistical difference. But for the unaffected side, you could see that there is a statistical improvement on the affected side, but the unaffected side remains the same. That means they are not using any compensatory mechanism. So there was an improvement. In fact, the gait stability ratio also reduced, indicating that their tendency to fall also improved. In fact, it reduced the tendency to fall. So we were doing all with our gait rehab system. We felt that, okay, what happens to the clinical parameters? These are the standard tests, the 10-meter walk test, the time taken for a person to get up from a chair and start walking. This is timed up tuck test. These are very well-known tests. And also what happened to the balance, overall balance. These are Berg balance scales. And we could see a statistical improvement in majority of the parameters. And so overall, our adaptive system literally improved one's gait in different dimensions. May it be the walking speed or the balance or the improvement in stability, so that there was a fall reduction, and also they in fact moved to a community level walking, where they can literally go out of their homes on their own, and they can get their own community life, enjoy their own community life. Of course, a lot of future scope still remains. We want to carry on with a larger post-stroke group, and of course with different variations in their disability. And also we are thinking of adding other modalities as well. So here we have taken mostly the energy consumption. We have not taken care the other gait parameters, but we have seen gait parameters as an indicator of how much improvement they have achieved because of the gait exercise. We are planning to add those as well. And also we have mostly given challenge based on the speed in our case. What about you ask them to walk uphill or downhill? That means we can increase or decrease the elevation of the treadmill, and because then that is the time where they will spend more energy. So that is what we plan to do in future. We are almost to launch this product as well, and we are already hearing good interest from the industrial community who would like to take this forward for licensing. I'm deeply grateful to my students who have graduated from my lab and also who are working along with me now. And of course, different funding agencies, the IISc Tide, Indo-French, I do also international projects, BIRAC for DBT, DST A-Star, different projects I have done, and also a very good thing, this year I have been chosen as a Tata Innovation Fellow as well, so I'll be getting good funding to carry on my work as well. These are the references, and thank you. This is the view again. So the first view was a sky view of our campus, and the last view is for the hostel. So please stop by whenever you are in Gujarat. I would be happy to welcome you. Thank you.
Thank you so much, Dr. Lahiri, and congratulations for the Tata Innovative Fellowship.
Thank you.
And that was such a cool talk. You started the talk by a disclaimer saying that you're not a neuroscientist, but I must say that such cross-disciplinary approach is what is required to tackle problems in neuroscience. And thank you so much for doing this amidst your hectic class schedules, and thanks a lot. And I think we have a few questions. Yeah,
thank you for this opportunity. Yes, please.
So we have a few questions. We'll jump into them right away. Abhilasha has a question whether can the insole gate data be collected in along with some other physiological data like EEG or ECG? If so, how are data packets from two or more streams synchronized in time?
Yes, wonderful question. So in fact, we are using at 200 samples per second. That is our gate data, and rightly said, EEG can be 1000 samples per second as well. But we are using, we can very easily synchronize them because we are also creating our data acquisition systems, and where we are in fact now interfacing it with many other devices as well. So it's very much possible.
Okay, yeah. We have a question from Abhishek Chari. Do you think it would be useful to add motivating stimuli into the VR environment? In essence, gamifying it to increase patient compliance with increasing resistance in the treadmill exercise platform?
Yes, yes, absolutely. That's a very good thing. So you have two questions, in fact. One is the gamification and the other is the resistive training. So very good questions. So the gamification we are doing, we are giving many motivational statements and motivational feedback like stars, like sounds, so audio, visual, in fact, verbal also we are giving through our system. And regarding the resistance, it's a very wonderful thing. So we are giving resistance in a very different way. So what we are trying to do, if a person's heart rate, say the energy consumption is increasing, and we see that the energy consumption is going towards the threshold, instead of reducing the treadmill speed, we keep the same speed, sort of giving them a resistance and allowing them to improve themselves so that if they can learn and get adaptive to the increased speed, then only we increase the speed further. So we are giving them resistive, not by holding them back, which is also which is a constraint-induced movement therapy, which is something very frequently used in conventional settings. We are giving constraint in a very different way, sort of giving them the increased challenge and allowing them time to improve their abilities.
Okay, great. Yeah, thank you so much. And I think that's the questions, and thanks a lot.
Wonderful. Thank you.
Thank you so much.
Thank you. Thank you.
And to all of you who had asked questions to Dr. Neha, she has answered them in the chat, in the Q&A box. Please access your questions, and she has very patiently typed all her answers for all of you. Thank you, Dr. Neha. Thanks a lot for this. The next, I'd like to hand over the mic to Dr. Sulagna Das. She is the co-host, and she'll take on from this point. Over to you, Sulagna.
Thanks a lot, Bhavna. And I will be moderating the next two talks for this session. So we'll start with Dr. Hena Firdaus, and we would like to extend a very warm welcome to her. She's an assistant professor at the Central University of Jharkhand, where she and her team work on understanding the genetic mechanisms of neuromuscle development and functions by using classical and molecular genetic tools. She has won many oral presentation awards in conferences such as the International Conference on Agriculture and Environmental Biotechnology in 2018 and the Indian Youth Science Congress. Today, she is representing the neuronal development field and will give a talk on understanding flight muscle functioning by employing forward genetic approaches, which help in deciphering the role of various structural proteins in these muscle functioning in Drosophila. Dr. Hena, thanks a lot for accepting our invitation to speak in this conference, and over to you.
Okay, thank you, Sulagna. Am I audible?
Yes, we can see your slides. You're perfectly audible. Thanks.
Okay, fine. Okay, so the title of my talk is Genetics on the Fly, Deciphering the Role of a Regulatory Protein in Flight Muscle Development and Function. Over a decade of research on Drosophila mesodermal specification, it has highlighted the role of twist, a major regulatory transcription factor in specifying the mesodermal cells early during embryonic development. Now, these mesodermal cells, they receive inductive cues from the overlying epidermis, and by the cell autonomous expression of various patterning genes, they are subdivided into various lineages like somatic, cardiac, visceral, and fat bodies. And these somatic cells then undergo asymmetric division, giving rise to embryonic founders, competent myoblasts, and adult precursors. These embryonic founders, they fuse with 3, 5, or 30 competent myoblasts, seeding each muscle fiber. So you can see here a picture of a body wall muscle of Drosophila embryo, and you can see each hemisegment has a set of 30 muscle fibers seeded by 30 embryonic founders. The other precursors which are specified during this embryonic stages, they remain quiescent, they attach to the neurons and imaginal discs, and they get reactivated during the pupal metamorphosis, giving rise to adult set of muscles required for adult life, for example, walking, jumping, or flight. Okay, so the model system which we are working on is Drosophila indirect flight muscles. Now, this is a flight muscle which indirectly powers the flight, and it consists of two groups of muscles, DLMs, dorsolongitudinal muscles, which are highlighted here in blue, which runs from the anterior end of the thorax to the posterior end. And another set of muscle is DVM, which is a set of 3 muscle fibers running along the dorsoventral axis. Now, these two muscles, these are the polarized light images of these muscle fibers, DLMs and DVMs here. These two muscle fibers, they contract antagonistically, and they bring about the thoracic deformation. And this thoracic deformation via the cuticle is transmitted to the wings, and this brings about the wing movement, up and down wing movement, and the flight in Drosophila. Now, if you look at the ultrastructure level, Drosophila flight muscle is quite comparable to that of the human skeletal muscle. Though human skeletal muscles are definitely much larger, comprising of thousands of sarcomeres, whereas a stretch of just 300 sarcomere is sufficient to build up a myofiber extending from one end of the thorax to the other end of the thorax. So at the structure level, ultrastructure level, the proteins which forms the sarcomere, which is the basic structural and functional unit in muscles, they are conserved in fly, and it consists of the thin filament and its associated proteins, the thick filament, which is majorly myosin heavy chain, and the Z-disc, which attaches this thin filament and brings about the contraction, muscle contraction. The muscle model, which we work on, the indirect flight muscles, it's a very good model for studying muscle development and function. Why? Because it's one of the largest muscle present in the fly body, and it's the only fibrillar muscle, which is quite comparable to the human skeletal muscles in its basic structure. Physiologically, if you look at, these muscles are similar to cardiac muscles in calcium and stretch activated contraction. IFMs are not required for the fly's survival. So if there's any degeneration of IFMs, the fly survives and allows us to do study. There are many IFM specific structural protein isoforms for which we can create null mutants and study the functions there in that background. And of course, all the steps during myogenesis, they are well characterized and it can be followed in real time using time-lapse imaging. And we can also study the influence of nerve and tendon cells during myogenesis, making IFM a very good model for studying muscle development and function. Now, in 1970s, people were engaged in doing classical genetic screen, which was based on the reverse genetics approach, where they were doing behavioral screens for flight muscle defects, and then they were mapping the gene which was responsible for this defect. And one of these mutations which was generated during these 1970s was flightless H. In short, we call it as FLE-H mutation. It was an EMS it was generated by the classical EMS mutagenesis screen. And the work which I'm going to show you is basically this EMS, this FLE-H mutant, which we had characterized in the lab. We procured this fly line from the stock center, and we started working on this. So the first thing we did was to check whether this mutation showed flight defect or not. And we found that this mutant was completely flightless. And when we looked at the muscle, the indirect flight muscles, we found that this muscle was completely degenerated in the adult flies. This picture here shows a normal jump muscles, whereas the 6 fibers of the DLMs, they have undergone
Dr. Firdaus, can you hear us? Hello. I think we lost you. Okay, sorry. So it seems like we just lost Dr. Firdaus for a little bit. She's reconnecting, as I can see. So just please hang on for a minute. Hopefully she'll be back soon. Okay, I think she is connected. Dr. Firdaus, can you hear us? Maybe you can log out and log back in if that's easier. Okay, I think that's what she's doing. No, she's still here.
Hello.
Yes, okay, now we can hear you. Just have to unmute yourself. You're still muted. Yeah,
can you hear me?
Yes, now, yes.
Okay. I was on this slide?
Yes, you were on this slide, exactly.
Okay.
Yeah, now
so
we hear you. Nice.
Okay. So this FLE-H mutation showed flightless behavior. And when we looked at the IFM muscles, we found that it was completely degenerated. And this here you can see the jump muscles on the thorax, which is normal. And this mutation was rescued by another allele of MHC, myosin heavy chain, which harbored a mutation in actin binding domain, which did not allow it to interact with the actin, and thus not allowing actomyosin interaction and no muscle contraction. And FLE-H was also rescued by another construct, which was MHC 10, which is a null myosin, and we're expressing another construct of myosin, which is a headless myosin. So actually, basically, these two constructs of myosins ensures that there's a reduced actomyosin interaction or reduced contraction force. What I mean by reducing contraction force is that under the normal scenario, the myosin rods, the heads region, they make interaction with the actin and they slide over the actin filament and bringing about the muscle contraction. However, in the Y97 construct, this head region is missing. Hence, the myosin rod, though it assembles normally, It cannot make interaction with the actin, and there's no actomyosin interaction or sliding, and hence no muscle contraction. So this mutation could be rescued by a construct which did not allow actomyosin interaction, suggesting that reducing contraction force could rescue the FLEH mutation. So then we went ahead and did several of other behavioral experiments. And the first one was, since at that time, many of the mutants which they had isolated had a temperature sensitive phenotype. So we brought this FLEH mutation, raised the flies at lower temperature, that is 18 degree. And then we did the flight test and we found that they had a gradation of flight phenotype. And the muscle was completely normal as compared to the FLEH mutants, which were raised at 25 degrees Celsius, suggesting that this mutation had a temperature sensitive phenotype. Also, this mutant showed associated lethality dispersed over different developmental stages, and mutation also affected the working of other muscles which were involved in walking and jumping. So here the jumping was reduced as well as the walking of the adult fly was reduced, suggesting that the walking leg muscles and the jump muscles were also affected by this mutation. Then we went ahead and looked at the developmental profile of the FLEH mutants, and we found that the FLEH in the DLMs, the dorsal-longitudinal muscles of the IFM, the development was completely normal, and this shows the sarcomere organization here. This is staining for the actin, and this is staining for the myosin, and this is the merge. And it showed that during pupil metamorphosis, the development was completely normal till 75 hours puparium formation, and it was only during the adult stages that the degeneration of the sarcomere structure was happening. And we know that the first twitch in the muscles happens at around 75 hours APF, suggesting that uncontrolled actomyosin interaction at post 75 hours APF could lead to the degeneration of the DLM fibers in the FLEH mutants. This is again the vertical section of the muscle fiber in the mutant background. This, the red dots here, they are the individual muscle fiber, myofibrils, stained by the colloidin, which stains actin. This is for the myosin, and this is an overlap. And it shows here that the organization of the myofibril is disrupted in the FLEH mutation, but it is not, it is slightly rescued at the mutation in the mutants which were raised at 18 degrees Celsius. Then once we had done the behavioral and phenotypic characterization, came the Herculean task of genetic mapping and rescue of FLEH. And then we went ahead and did recombination mapping, deficiency and duplication mapping, and we could place our mutation in the 16F cytological mark position. And the only known gene which was present at 16F was troponin I. And troponin I, so we then did complementation with one of the mutations of the troponin I, which was held up 2. Now this HDP2 mutation, it harbors a mutation in the coding sequence, and these held up 2 mutants also show muscle degeneration phenotype. FLEH was not able to complement held up 2, suggesting that FLEH mutation could be in that particular gene. Then we mutation with the L9, troponin I, L9 eyes were functionally all right. And they also showed the completely normal sarcomere organization. Then once we started sequencing, we could not find any mutation in the coding sequence, but upon sequencing the upstream regulatory region, we found certain changes. So you can see here the changes in the upstream regulatory sequence. So we could place the mutation FLEH in the upstream regulatory region or promoter of the WPA gene, which codes for the troponin I. Now, troponin I is one of the key elements of the troponin complex, which regulates the muscle contraction. So then we looked at the effect of the mutation at the molecular level, and we found that in this mutants, the levels of the troponin I is significantly reduced. However, at the 18 degree, we found a slightly comparable to that of the control flies. The protein levels, protein accumulation was also much lower in the FLH mutants raised at 25 degrees Celsius. Then we went ahead and did an EMSA, and with one of the key transcription factor, which possibly had the binding region at the mutant sites, and we found that the Drosophila myocyte enhancer factor 2 binding was abrogated in the mutants. And then we did the promoter assay in the C2C12 cell lines, and we found a relative reduced luciferase activity in the FLH mutants, suggesting that the promoter firing was not to the normal because the Drosophila myocyte enhancer factor 2 protein could not bind to these mutant sites. What was interesting during our study was that there was a coordinated downregulation of the transcripts of other thin filament proteins. So troponin I forms a complex with troponin C and troponin T here. And two major isoforms of troponin C and troponin T in IFM is TNC4 and TNT10A. And both these transcripts levels were reduced. Also, the TM2, the troponiacin levels were reduced, suggesting that in the absence of enough troponin I, there was a coordinated downregulation of other transcripts of the thin filament protein. So to summarize, what I showed you is that the FLH was a mutation in the regulatory region of the VUPEV gene, which codes for the troponin I, and it leads to a muscle hypercontraction. Basically, muscle hypercontraction is a phenotype wherein the muscles are formed normally, but due to uncontrolled actomyosin interaction, the muscles then degenerate. So in troponin I FLH mutation, the troponin I transcript and the protein accumulation is reduced, and there's a downregulation of other thin filament proteins in the FLH mutants. So this is a schematic which shows here in the wild type scenario, there's a normal complex of the troponin I which is formed assembled at the actin filament, which regulates the actin and the myosin interaction. However, in the FLH mutants, these troponin complexes are not formed, which leads to the uncontrolled actomyosin interaction and thus the breakdown of the muscles because of the excessive stress or the forced generation. So with this, I'll end my talk, and I would like to acknowledge Professor Pendra of IISc, where I did this work. And if you have any questions, I'll take up those questions.
Thank you, Dr. Hena, for an excellent talk on elegant use of genetics to understand muscle development. I just had one question, and maybe there might be other questions which might pop up in the chat box, and if you could answer them later on. So I just had one question, like regarding you see this interesting observation of downregulation of the different components of the troponin complex. With the downregulation of the troponin 1, do you think that they're all regulated by the same transcription factor, or is there something else that's happening? Is it like some sort of a?
Well, actually, it does happen that many of the thin filament proteins of muscles, they are regulated by the same transcription factor. That's true. Okay. But what is interesting will be interesting to know how they sense their levels. How do they sense that one is not there, one component is not there of the complex, and then the downregulation of the other components happens? That would be interesting to study in the future.
Yeah, that's a really interesting question. Okay, thank you so much. And I would like to request you if you could check the chat, the question and answer box later on.
Yeah, I'll do that.
Okay. Okay. Thanks.
Oh, thank you.
Thank you so much. And we will now move on to our last speaker of this scientific session. And we would like to extend a very warm welcome to Dr. Devika Narain. She's an assistant professor at Erasmus Medical Center in the Netherlands. And using combined approaches that include mathematics, neurobiology, machine learning, and behavioral analysis, so using this whole gamut of approaches, she and her team work on understanding the neural basis of temporal control and how sophisticated timely behaviors are generated. She received the 2020 NWO VD grant and is also a recipient of 2018 Marie Curie Individual Fellowship. Today, she is representing the systems and computational neuroscience field and will give a talk about understanding the neuronal dynamics that enable control of timed behavior using multi-scale computational models. Dr. Devika, thank you so much for accepting our invitation to speak in this conference, and over to you. Looking forward to your talk.
Thank you, Sulagna. I hope you can hear me.
Yes, we can hear you perfectly. You can see the slides as well. Thank you.
Excellent. So let me start by saying it's been a crazy year for all of us, and hope everyone out there is well, and hope the next year brings good things for us all. And of course, let me start by saying a big thank you to Shruti and to Vaishnavi. I mean, this is a fantastic initiative, and I'm very grateful to be part of it. So yeah, the organizers did ask me to start by talking a little bit about my background. And I was thinking, since we're running a little bit late in this session, maybe we could see later if we have time, and I'll get back to that. So let me jump into the science of it all. I study, as Sulagna very kindly introduced, the temporal basis, so the neural basis of temporal perception. And today I'm going to just talk about this one specific discovery. And this is largely following from my postdoctoral work. Okay, so let's see. So what do I mean by control of timing? Well, let me give you 3 examples from daily life. And let's, of course, start with our national pastime, which is cricket. I hope that still remains the case. And I don't know if you've had this experience, but we've heard a million times over commentary that cricket is all about timing. And here we see a batter. Batters must show the flexibility to control the timing of their movements in order to time the ball correctly. The second example is a regular traffic light in the Netherlands. I don't know if you know, but there are a lot of bicycles in the Netherlands, and they have their own dedicated traffic lights. So that's what you see over here. And here an example would be that if this light were red or if it were yellow, that would determine the urgency with which you would apply the brakes. So you would have to show some flexibility based on the context in the environment about how much time you have in order to stop the motion of your automobile or your bicycle. And a third example of temporal control is for any of you who've ever had music lessons and your music teacher complained about that you rushed through this piece, you have the ability to deliberately change the tempo of the rendition. And that is temporal control, that our ability to change on cue deliberately the timing of our movements. So that is specifically what we're trying to get at today. And I want to discuss the neural mechanisms that could potentially cover these kinds of behaviors, but specifically cover this one experiment where we try to study this question in a controlled experimental setting. So today we covered flies, we covered I suppose humans, patients. We ran through a lot of different topics, and I guess I'm going to end this with monkeys. So we trained monkeys on the following task. Monkeys were seated in front of a screen, and they would see a color. So just like the traffic light, except that they would see 2 colors. Either it would be red, or if it would be blue. If the cue color was red, the monkey they would see a flash of light, and if it was red, they would wait for 800 milliseconds. and then they would just move their eyes. So there's no motion during this duration. It's a delayed saccade or a delayed button press. So that when this is a circle, they have to move their eyes after, let's say, 800 milliseconds. And when the cue is a square, their finger is on a button and they just have to press it after 800 milliseconds. Now, if the cue were blue in color, they would do the same task, but this delay would be longer and they would only get rewarded if this delay was 1500 milliseconds. Now, the catch is that on any given trial, these 4 conditions could be randomly sampled and the monkey would have to correctly use the effector that is specified by the cue and at the right time. So this is what we mean by flexible control of timing from external cues. Now, this is just to show you that monkeys can actually perform this task. Let me keep one eye on the watch. So on the x-axis, what you see is the production time. This is the time at which the monkey is responding. And what you see here is on average, monkeys are quite close to the 800 millisecond mark. And on average over here, across an entire session, 800 trials, monkeys are quite close to the 1500 mark as well. And this applies to, this is another monkey's responses. One thing that we do see is that there is a lot of variability in these responses. So you can see that the histogram is quite spread out. So what we're now going to do is we're going to harness all of this variability by breaking up these responses into smaller time bins. So here you see, I'm going to break this up into a lot of smaller red bins and blue bins here. And so every time you see a bunch of red lines, they belong to the short interval. If you see a bunch of blue lines, they belong to the long interval. Now, while the monkeys were performing this task, we were recording from the medial frontal cortex, so somewhere around here, of the monkey, using using silicon probes and large-scale electrophysiology. So here you see an example of a raster of a single neuron, and this is aligned to the time at which the monkey moved its eyes. And it's sorted from the longest response time, the trial where the monkey made the longest response time, to the shortest one. So it's just sorted like this. And these are the bins that I was just telling you about in the previous slide. So what we're now going to do is align everything to basically the time of the flash. And we're going to extract for each of these different colors an approximation of what is the rate at which this neuron was firing, and that's what you see over here. So you see here for the short condition, this is the rate at which this particular neuron was firing, and the same neuron for the longer condition, you see these blue lines, and that's obtained from this graph over here. All right. So let me start by giving you an overview of the kinds of properties that we found across the firing rate profiles of these neurons in this area. The first thing that you see is so first of all, there are 4 neurons over here. In the last graph, I showed you the red curve on top of the blue curve. All I've done is, so notice that the y-axis is exactly the same. All I've done is I've just separated, I put the red curves on top of the blue curves. Okay, so it's exactly the same y-axis. And it'll become clear just in a moment why I've done this. So this is the firing rate of these 2 conditions. This is a single neuron, second, third, and fourth neuron. So the first thing that really pops out at you is that each of these neurons is doing something entirely different. And this is what we would call heterogeneity. And that's very common in the frontal neural regions. The second thing that you see is that these neurons are not simply going up or down. They're doing something highly nonlinear. So they have a complex pattern of activity. But it's the third thing that probably struck us the most and is perhaps the most interesting finding of this work. If you now select any of these 4 neurons and take the endpoints of the red curve and then let it remain fixed on this other end. So take the endpoints of the red curve and stretch them out till they are the same length as the blue curve, or take these endpoints and stretch them out till they're the same length as the blue curve. And what you see is that these neurons are stretching and compressing their own activity profile. They have a compressed activity profile for the short interval and an expanded dilated profile for the long intervals. And here are 4 more examples of that phenomena. And so this is a phenomena that I'm going to talk about for the rest of the talk. And this is something we call temporal scaling, which is the stretching and compressing of a neuron to encode different time intervals. So what we would like to do now is these are just 8 neurons. Is this representative of what is going on in this entire brain region? So for that, we first need to quantify this phenomenon. And we're going to do it in a very simple way. We're going to do exactly what you guys just did. We're going to take the red curve, stretch it out, and compute an R-square with the blue curve on average. And that R-square, we're going to call scaling index. So if this value is 1, the neuron is scaling perfectly. If this value is 0, then the neuron is not scaling at all. So what we can now do is we can use machine learning techniques to kind of get an idea about what is the entire population potentially doing in terms of this kind of scaling index. And what you see here on the x-axis is the scaling index. And what you see over here is you should consider a 3-dimensional histogram, where the yellowest part of this histogram represents the peak of this curve. And that peak over here lies between 0.5 and 1. So we could consider that on the whole, this entire population, this population of 522 neurons, largely has its peak. There's a distribution whose peak lies on the high side of the scaling index. So I'm going to use this analysis metric to introduce you to to the surrounding neural circuit. So right now we were talking about the medial frontal cortex, the frontal cortical region. Now so this area projects to the basal ganglia, and specifically to a part of the basal ganglia called the caudate. And here also we recorded from 509 neurons, and we did exactly the same analysis on it. And this should not be surprising. If this area really projects to the caudate would inherit the same pattern of activity. And therefore, it's not surprising that we find that the peak of the caudate neurons also lie on this high end of this spectrum. Now, what we can also do is we can identify antidromically. So we can stimulate here in the medial frontal cortex, and we can ask which are the thalamic neurons that are sending inputs specifically to this area. And then we only do the analysis on those input neurons. And what we find is that these thalamic inputs, the afferents that are going to the medial frontal cortex, have a very different distribution, fundamentally different from the MFC. And here you see that the peak of this distribution lies between 0 and 0.5. And that really made us ask what might be going on here. Could it be that these inputs, which are not scaling themselves, play an interesting computational role in enabling scaling in the medial frontal cortex? So to understand this better, I'm going to switch to an engineering framework, actually, and I'm going to take you through this. So here, imagine R is a variable that represents the rate, the firing rate of a single neuron. And we have an entire vector over time of firing rates of n neurons. And at any given time, this vector, I'm going to call X. So this is the vector that is representing my entire population at any given point of time. And this is evolving over time. That's why it's suitably a dynamical systems perspective. So what we're going to do is we're going to ask, what is the rate of change of the evolution of this particular vector? And that we're going to assume that it depends on 2 factors. The first factor is the local connectivity of a certain brain region. And the second factor is any external inputs that might be influencing the dynamics in this particular area. So just to spell it out a little more for you, what I mean by this is that I'm going to make this implicit assumption that the cortex, this term is modeling basically the local interactions of the neurons within the cortex, which are very recurrently connected to each other. And this term over here, which is right outside, is representing the non-scaling inputs from the thalamus that are potentially having some effect on these dynamics as well. And one thing I want to point out is that these monkeys were trained for 2 years. So there's no more learning going on in these circuits. We assume that the weights, the synaptic weights and the learning of the task in this brain region has crystallized. So these are important assumptions. And one way we could study this, I mean, if you were here, I think on Friday, Kanakarajan, who actually pioneered many of the original techniques in recurrent neural networks, gave a very nice talk about her new applications in her lab. So we just used recurrent neural networks as a tool to ask ourselves, what kinds of inputs do we need to give a network under this framework that I just described to you, in order to get the performance that the monkey gave us, and to reproduce without fitting any data. This is entirely, the network has never seen any data. We just want to understand what are the inputs, external inputs necessary to induce activity in this model that would cover all of the traits that we discussed: heterogeneity, nonlinearity, and temporal scaling. And the answer was that we require graded inputs. So what this means, it's like a step function. It turns on and then it roughly stays on. And it for different cues, this exhibits different levels. So when the advantage of having a recurrent neural network as a model is that we can open it up like a black box. You can open it up and you can look at what do the dynamics look like in low dimensional spaces. And these are better interpretable than neural data. So what we saw over here is that the network was arranging the activity along a single dimension. And if you looked at the speed of the trajectory along these different dimensions, you would see that the speed is changing. So the longest interval would be the slowest, and the shortest interval would be the fastest. So just to give you, let's go deeper into this thought. I suddenly started talking about speed as this is what we got out of the network. So now let's do a thought experiment very quickly, just to understand this a little bit deeper. Imagine you are in a space where every dimension represents the activity of a single neuron. So neuron 1, neuron 2, and neuron 3. So now the time is implicit here. There's no axis that represents time, but it is implicit because let's say this is the moment where the flash came in and the monkey has started timing. The activity of all these 3 neurons will start evolving for these 2 conditions. And then the monkey makes a response. So now my question to you is, imagine that these 3 neurons were scaling perfectly. So when you stretch out the red curve, it fit completely on top of the blue curve. Under those circumstances, what would you expect this trajectory to look like? And I don't know if you're typing stuff on chat or thinking about it, but in the interest of time, I'm just going to tell you that if these neurons were scaling perfectly, we would be able to find a subspace where if I projected the activity of these trajectories, they would move along the same path. And because these are representing 2 different time intervals and they're moving along the same path, that necessarily means that they're moving at different speeds. So we wanted to understand in our 500 dimensional space, what is the right way to find this gray subspace? So right now it's easy to visualize in 3 dimensions. We can say, okay, there's a plane, but there's probably a hyperplane in 500 dimensional space where we would be able to find this kind of scaling activity. And so we developed, you might have heard about this technique called principal component analysis, and analogous to that technique, we developed a new technique called scaling component analysis, where we try to find this hyperplane and project the activity of these neural trajectories onto this hyperplane. And if we had done this correctly, then there are 2 predictions. The first is all of these trajectories should be moving along roughly the same path. And second, from the speed of these trajectories, I should be able to decode quite accurately the time that the monkey produced in the actual behavior. So that's what we find. We find that they're moving roughly along the same trajectory. And these are 40 millisecond bins. These are 80 millisecond bins. And I can very well, to this precision, I can decode the time that the monkey produced. So this really is something that we could look at the neural population and really, you give me the speed of the trajectory, I will tell you the time that the monkey produced on average. So what did we get out of this? We understood that these two computations are just two sides of a coin. The input is organizing trajectories along a dimension, which determines the speed at which they will evolve. But if I rotate the same subspace, I find that all of these trajectories are also moving along the same path at different speeds. So, this is, we would have never uncovered this without the network perspective. And then we could do the same analysis on the thalamic population. And we could see that, okay, there is really a completely different structure in the thalamic population data compared to the MFC data. So that allows us to understand this a little bit better. And then we wanted to understand, so what is so special about this graded input that it changes the speed of the neural trajectory? Now, I'm not going to go into details about the mathematical model, but I just want to give you the intuition behind it. And the answer is very simple. If you increase the input of a neuron, the output of the neuron does not increase linearly. It kind of saturates and non-linearly plateaus. So if I'm over here, if I have a unit change in activity, I get a unit change in output. So that's the dynamic range of the neuron. But if I'm over here, unit change in the activity will give me less than unit change in the output. Now, that means that the responsivity of the neuron, the sensitivity of the neuron, and if you are from a technical background, it also means that the eigenvalue of the entire system is going to change. So the time constant at which the neural activity, the dynamics is evolving is changing. And that means that just by changing the amplitude of the input to this system, I can change the time over which the activity of the same neuron will unfold. And so that is our explanation for what might be happening behind this phenomena. So just to summarize, what we're saying is that at the population level, it's the speed of the dynamics that is ultimately changing at the population level in order to produce different time intervals, and that potentially subcortical inputs from the thalamus change in magnitude and interact with non-linearities in the cortex in order to change the time scale over which these neurons, this neural activity evolves, thereby changing, basically thereby causing the stretching and compressing of the neural profile. So I just wanted to, have plenty of time for questions. So I wanted to keep it short. And this work was done at Mehrdad Jazairi's lab. I was doing my postdoc with him in the US. And of course, science always gives you the opportunity to work with remarkable people. And I was very lucky to be on the same team as Jing. And of course, now I just started recently my own lab in the Netherlands. And if there is time, or maybe in the breakout room, we can address what should the innovation that we want to do, which is talk about, academic journeys and changing fields and all of that, what it is like to leave India and what lies beyond. So if you're interested, I look forward to having that conversation. And with that, I'm going to hand it back to Sulagna. So yeah, there you go.
Thanks a lot, Dr. Narain. That was an excellent talk. And this concept of temporal scaling, I think, I just had one question. Do you think it is also dependent on the intrinsic excitability of the neurons, how they would scale?
That's a great question, actually. So this is a within neuron phenomena. So if you, let's say there is heterogeneity across neurons in intrinsic excitability, but given the same baseline firing rate of a single neuron, you would observe this phenomena within a neuron. So it's a within neuron measure. Therefore, even with heterogeneity in the intrinsic firing rate, you could still observe it orthogonal to that effect.
Okay, Thank you. So I think there might be more questions, but since we are going to the breakout room right after, people can ask you the questions right there, and maybe there might be some in the chat box as well, which will relate to you. So just wanted to take a moment to thank all the speakers for a series of excellent talk and presenting all these multidisciplinary approaches to tackling all these important questions in neuroscience. So thank you all for your time and presenting at NeuroFemIndia. We also wanted to thank the audience for being so patient and engaging. And we just wanted to request all the conference attendees who are interested in joining the breakout session to interact with our speakers and to use the Zoom link that has been added to the chat box. Also, once you are in the breakout session, please let us know which speaker you are interested in talking with, and we will accordingly put you in the same room. After those discussions, we will be returning back to our public webinar forum again at 4:30 p.m. IST for the Ungender workshop on gender awareness and biosensitization. And members of the audience, we would like to request you to use the separate Zoom webinar link for this event that has been emailed to you separately to re-enter the conference for the Ungender workshop. So hope to see you all at 4:30 p.m. and don't forget to join the breakout rooms. Thank you all.